So we were discussing Golgi body. In that we saw a lot of aspects. We saw dictyosome, which is present in plant cell. Golgi in plant cell known as dictyosome. So let's start with the basic information, more information. So what if I ask you how many cisternae? Cisternae is this kind of structure you see, one, two. So how many cisternae would be there? That's the question to you first. But still, if you don't know, I will tell you. There are like from 4 to 8 you'll find many, but if you talk about some other protists, you can also find 60 in them. But when we talk about the human being, like in animals or in plants, you'll find 4 to 8 cisternae, not more than that. This is cisternae. Do not pronounce this as Christie, because Christie is a structure which is present. This all that is also unfolding, but that's present in mitochondria. Okay, and in protists you'll find more than 60 as well. So this the structure. Cisternae is this kind of structure, and if this the one cisternae, the space, the empty space you see is known as cisternal space, or we also call this as a lumen. Lumen is a good name because in all organelles you find empty space, then an empty space known as lumen. Lumen is a cavity. So here the cavity is known as lumen. Lumen would be called, but this is cisternae lumen, so we call this as cisternal space. Okay. Then a stacked group of small flat sac cisternae is called Golgi complex, which I discussed. That let me move a slide. Here we discussed that when you find one cisternae sac, here's the one cisternae sac, that is Golgi body. When you find a bundle of stack together, that is Golgi apparatus. When you find more than these structure and they are associated with vesicle. Vesicle are like these all dot-like structure present on the entire these. That is known as Golgi complex. It was done. Then this was discovered, and this all basic information was done. Okay, so now let's uh, let's discuss more about cisternae. You'll find cisternae. Some cisternae has phases, which I'm discussing. I will discuss this. I'm supposed to discuss from starting this because you want understand this again. So let's start. The Golgi has 3 components. Golgi has 3 components. You will find 3 components. First is known as cisternae. Second structure which you will find is known as medial phase, not middle, but medial phase. Then in the third you'll find trans phase. So what does these 3 mean? These 3, first is, let me just go towards the structure. Here is the picture. So here's the picture. You'll find first thing here, this organelle is present near the nucleus. Of course, near the nucleus that's Golgi, not Golgi but ER. But after that you'll find this Golgi. This is the Golgi. These are the ER, SER, RER. Here this is the nucleus. Okay, first thing. Then the dot-like structure you're finding is the protein which is present on RER. RER is the ribosome. RER, ribosomal ER, endoplasmic reticulum. But the structure where you don't find ribosome is known as SER. This should be very much clear to understand Golgi. Then see, this SER will generate a vesicle, and that vesicle will go to that these will fuse together. When they fuse together, they'll find another structure. They'll make another structure. That structure is known as Golgi body simply. But when this fused form of the vesicle comes, that is accepted by one thing that is known as cis phase. Cis phase is the first phase of Golgi body. Okay, this have many names.